Okay, good morning, everyone. Welcome to this free webinar. Today's webinar is going to be dedicated to the program called Omicslogic Data Science. And Omicslogic is a company that brings together participants from all over the world. Welcome to this free webinar. Okay, one second. Okay, sorry about that. I had to unmute the other one. So let me resume. So Omicslogic is a training program developed by Pine Biotech, which has been started as a technology transfer company from the University of Haifa, from the Tauber Bioinformatics Research Center. There at the center, the team is focused on developing novel algorithmic approaches to multi-omics data integration. And we have a team that works on translating that research platform to practical use in many different areas of molecular medicine, genomics, transcriptomics, metagenomics, and other types of omics data. But as a result of this, we have developed a program that is now being used by thousands of people from around the world and multiple universities. And as a result, we have trained a number of students and researchers to utilize some of the resources to elevate their level of bioinformatics and provide with excellent support to use big data in various areas of research. So Omicslogic is essentially a program that provides training in bioinformatics that enables independent research that is guided by mentors and peer example. And we are developing a growing community with shared passion for data-driven research and appreciation of citizen science. So not only researchers that are already doing research, but also those that are interested to do that and are interested to do that even without a deep expertise that they already possess. And our goal is to enable students, clinicians, and faculty of all backgrounds to develop novel and independent research using the latest technology in life and data science. And so what we've realized over the course of these programs is that data science plays a central role in the ability to extract meaningful insights from biomedical and biological data. And as a result, we identified the need to provide training in statistical analysis and machine learning, design of experiments, how to properly design them to get the right kind of information, including elimination of technical variation, and provide tools and skills in data mining for meaningful patterns and associations between phenotype and genotype at multiple omics levels, and to study complex biological systems using big data. So the goal of this data science program is to really introduce the participants to various types of omics data, including different technical formats and data types and file formats, and what are the specific challenges to translate those raw data formats to structured data formats, to learn about exploratory data analysis and the steps of processing data in preparation for downstream analysis. So how to actually use the information that is structured and prepared, and what kinds of considerations we should have before we start the analysis itself. Introduce the participants to big data analysis tools for large-scale project data, and to try different methods of machine learning in R and Python, and then to learn the important links between data and biology. So how to interpret the found patterns, linking the methods that were used to analyze the data with biologically meaningful insights. Who is this program for? So the program is really for anyone with an interest in biological data, and specifically in omics data, including students from high school, colleges, universities, but also for biologists and clinicians that have some kind of research work that is probably acquiring more and more characteristics of a data science project. And what is a data science project, essentially? A data science project is a project that requires in-depth understanding of data, how it was generated, what does it represent, what is the context of the project itself. It also requires to wrangle and process that data. So oftentimes we start from a dump of data, whether it is raw sequencing reads or all kinds of notes about something. So it's really unorganized information. And the final part of it is that there's a challenge, and the challenge is to work through all of this mess and translate that into something meaningful. And meaningful can mean visualization, so that the visualization, visually the data is summarized in a proper way. It could also mean that we are going to take some biological experiment that can be viewed through multiple different perspectives, and we need to understand what do all of these perspectives mean, and that means data integration. And also it can mean we can train some kind of a machine learning model to help us understand what happens in a new data set based on a data set we've already explored. So we'll talk about all of these components, but I think that all of them are relevant for students that are learning about this, or for practitioners, those that are already using this kind of data in their research. And to really introduce all of these topics, we have our team here, which is myself, Dr. Mohit Mazumdar, and Dr. Harpreet Kaur. But we also have one of the mentors that will be joining specifically to walk us through some of these technical elements of data science. And to introduce him, I would like to pass it on to Clinton Kunha. Clinton, welcome.
Thank you, Ilya. So I will introduce myself through another amazing PowerPoint. I'll just share my screen. Could I have permission to share my screen? Perfect. All right, yes. Perfect. Thank you, Ilya. So hello, everyone. My name is Clinton. I studied at Carnegie Mellon University for my bachelor's degree in biological sciences. I did an honors degree there, and I also did 2 master's degrees, one in molecular medicine, that's wet lab research, and another one in dry lab research, so bioinformatics. I've learned several skills along the way, such as Python, R, Linux programming, and I've won some awards in different institutions. I also have a software engineering diploma, and I love both biology and programming. So just a little bit about my background. My first master's was about studying a protein known as ST20-like kinase, or SLK. And this was in a cancer lab where we studied different cell lines, some mouse models. Essentially, we were working on characterizing SLK's role in apoptosis, cell cycle, and cell migration, some common cancer phenotypes. And some things that we found out were that it's critical in normal development, and a complete knockout's lethal. And it plays a role in new induced mammary tumorigenesis. So breast cancer in mice. So that could have a phenotype, a human phenotype for future projects. So these are just some results. If you're used to wet lab techniques, you might be familiar with Western blots and focal adhesion assays. So the aim was to find out what SLK the role of SLK, or ST20-like kinase, in mouse embryonic fibroblasts by testing common cancer processes such as proliferation, migration, and apoptosis. And finally, the result was that we noticed that focal adhesion turnover seems to be impaired. So I studied this at the University of Ottawa, where I did my first master's. So after doing my wet lab research within a cancer lab, I I felt like I wanted to learn more analytical skills to analyze my data set. So looking at the trend around me, I noticed that biology was heading towards a more data science approach, more where you have to analyze different types of data with more sophisticated software tools. So then I realized that let me transition to a more bioinformatics role, and I joined another master's immediately after my first um, in the University of Guelph. So I did a master's in bioinformatics, and some of the reasons why I did that was because of the changing dynamics of the field, my passion for both biology and programming. I did get into computer science at Carnegie Mellon for my bachelor's, but at the time I was more interested in biology, and I never really knew much about bioinformatics or computational biology. So I, because of that, I got exposed to different disciplines such as statistics, mathematics, engineering, computer science, something that a person who just did biology might not generally be exposed to. So it did give me a lot of variety, and I did enjoy that. So it was very, and I still enjoy it. So after doing my master's in bioinformatics, um, I came across, I met Ilya through one of his interesting webinars, and he was talking about his bioinformatics for precision oncology program, and we were discussing about the different aspects, and I had the opportunity to be one of the mentors for the program. I'm currently doing a teaching and training of adult certificate expected in August. So I do have the formal teaching skills to embark in such a program. And it was a very nice experience because I have the interest for cancer research, and it was very enlightening to see my students with their own research coming from a more wet lab approach, which is something I'm familiar with. So it was a very, I had the opportunity to talk to um, wet lab research scientists, give them my insights, use the resources that we have to help improve their research by incorporating important data science techniques. But for this program, it was particularly data science techniques for cancer research. But um, the program that we're talking about is more the important software skills for more generic types of research. So these techniques are very important. And being a person who transitioned from a wet lab approach to a dry lab approach, I can definitely appreciate those techniques. And this was a fun experience, and I look forward to repeating the same with you all. So that is, does anyone have any questions?
So Clinton, I actually do have a question for you. I'll represent the audience here. So my question is, how difficult was it to transition from learning about life sciences and biology to data science or to, other things that you've done?
So initially I thought it might be very difficult because there were some programming concepts like R was a bit new to me. I did a little bit of Python I did some Python in my bachelor's degree, but it was minimal. I think with every new experience, you kind of have, there's a learning curve. So no matter what you do, you will have some sort of learning curve, and most people have to go through it. Thankfully, being in a good environment, so being in this course, you'll have experienced mentors to give you that learning environment, just like my professors during my master's program. So I'd say in my experience, every challenge has a learning curve. It's preferable to have a good learning environment and good mentors to help you support you during that learning curve.
And another question that I wanted to ask was, how do you know when you've learned something? I mean, there are many resources online, right? You can just download people's code and, try it and things like that. I mean, how do you know that you've actually learned some analytical technique or method?
So um, because I am in the process and I have been an instructor, there is a taxonomy. It's called Bloom's taxonomy. So there are different levels of learning. So it depends on how much depth you want to go into. So you do have a point that sometimes you might not be sure if you learned something. And educators like to use concept checking questions, different types of assessment, whether it's coding assessments or just simple quizzes or multiple choice questions. It's usually good to display your work in a project of some sort. So that really shows that you've mastered the tiny puzzle pieces in order to create your own novel work. So that would be one of the higher levels on that taxonomy, versus just typing a print command, which just kind of shows you that you memorize the recall. So you have that recall stage. So ultimately demonstrating your work in a creative project, that would show like your ultimate understanding. So through a research project. So whether that's publishing or doing presentations in front of other experienced people, or creating some manuscript, or doing a poster. So yeah.
Okay, great. Thank you very much. And if you do have any other questions, please do write them in the chat box and we'll be sure to address those. And I briefly want to pick it up from where Clinton has left off to talk a little bit about what are some of the challenges and opportunities, especially applying this type of skill, the technical computational programming, data science skills to big data. So the first thing that I would start with is that, surprisingly, we have access to a lot of high quality data sets. Not only are these data sets generated by experts in excellent labs, the data sets continuously update. So we have new and more detailed understanding of different areas of life sciences through the lens of these data. For example, just in this pandemic, we have a tremendous growth, not only in the quantity and quality of data, but also the rate of data submission. So if you compare this to previous pandemics, like the SARS-1, Ebola virus, MERS, or Zika, this pandemic has generated so much data in almost real time and has made that data accessible to almost anyone that would like to. Now the question is not so much about data availability, but what to do with this data to extract meaningful insights. And that's where I think these skills really come together. So first of all, data could be of many different types. It could be imaging data, clinical data, streaming data, omics data, chemical, phenotypic. There are really many different types of data. But for all of them, one of the first challenges is to create structured data that you can actually work with. And so to do that, the first thing that we will talk about is how to take these different types of omics data, including phenomics, phenotypic data, genomics, variant data, epigenomics, different types of histone modifications, DNA methylation, transcriptomic data, gene expression, and things like that, and how to take that information and start processing it, creating structured tables. And those tables could come in different formats. They could be matrices with just numbers. They could be data frames with both numbers and different types of levels for factors or names. And now that we have this structured data, now the second challenge is really to be able to ask the right kinds of questions about this data, right? Nobody just takes data and structures it so that you can, visually see what's in it, right? Everybody works with data to be able to answer some important questions. And that's another part of data science that I think differentiates a data technician from a data scientist. The science is really all about asking questions that are supported by evidence. And so the data exists to help us extract evidence from the data set, but for us to have a relevant question, what we want to do in this program is to introduce you to some of those critical and important and interesting questions that could be asked of these types of data sets so that you can really utilize the data effectively. And so what we hope that this program can provide for you is a complete roadmap for data science that will be an application to biomedical research data, but it will include things like finding different data sets, learning how to explore and visualize these data sets to understand major characteristics of the data, how to understand different types of research methods that others use and extract the evidence that you need to answer a research question, and ultimately how to showcase or illustrate what you have found with relevant methods of insights so that you can visualize your data, so that you can identify the properties, the statistical properties to point to, and ultimately lead to understanding. So we call this program Omicslogic because really it combines the omics data and the practical skills with a logical approach. And it guides you from A to B, from knowing about all of these terms and wanting to apply them to actually understanding of how to apply them and then demonstrating what you have learned. So we'll take several different types of projects using biomedical omics data, from diagnostics in cancer biology, single cell and tissue composition, microbiome and infection. And in the context of these different types of topics, we will learn about your typical data analysis skills that you would have to learn to use effectively to extract meaningful insights, including data collection. So how was the data collected? What does it represent? Data cleaning. How do you extract only the relevant information or the good quality data, right? And make it tidy and ready for analysis. How to perform exploratory data analysis and how to iterate that process so that the insights come from the data, data-driven questions and hypotheses that you can generate, and then how to explore the data in a way that you can really apply statistical modeling and inference from those models so that you can get meaningful results. And obviously a big part of that is going to be focused on data mining and classification. So we will learn about different types of unsupervised and supervised techniques that have been used extensively on their own, but also as a part of meaningful bioinformatics work. And the principles of data science that include statistics and data wrangling are going to be very meaningful here when we go to machine learning, because you will essentially operate with an extraction from different portions of the data to train your model, to validate your findings, to make sure that your model is accurate. And you will learn to differentiate between different types of methods so that you can compare and contrast the results and select the ones that are most appropriate to the type of problem that you're solving. Now, the same methods are also included in many different types of biological data mining, and those include things like differential gene expression or principal component analysis and standard tools that are adapted from these methods to address more and more technical variation and complexity in the types of data that is being collected, including integration of multi-omics data, but also including more and more advanced data types like single cell data and data that combines multiple elements together. And so that's why we really are going to be looking at a variety of applications for multi-omics data integration and integration of this omics data with clinical data using the different examples that we will try. So to give you an overview of what this program is going to look like, I would like to take you to the program page. So for all of you involved, sorry, let me go over here. Okay, so everyone that is interested to join this program, the easiest way to show you what this program is going to include is to take you to the program page. So this is a program page. It is called LBRN Omicslogic Data Science. And so I'll just explain what does LBRN mean. LBRN stands for Louisiana Biomedical Research Network, our major partner in this program. And because of their support, we are able to make this program more accessible to more people. And so the types of information that you will get on this page are going to, I think, address many of the questions that you might have joining today's webinar. So first of all, the program is going to start on July 12th and last for a month. And during this time, we will cover a number of different topics by participating in live sessions like today, where you can interact with the mentors and get answers to your question, as well as practice in class to perform different types of analysis. You will also be able to use asynchronous resources in between our sessions to gain more technical skills at your own flexible schedule. So in many cases, we will go through a portion of the material by going through them in our session, and then we will point you to resources that you can supplement this knowledge with by performing actual technical steps of writing the code, trying to run the code, getting the outputs, and even troubleshooting some of your results. So to illustrate how that part of it will work, let me ask you, how many of you already have an account on Omicslogic? So please, if you do have an account, maybe you can put in number 1 in the chat. If you, okay, great, thank you, Leanna. If you don't have an account, just take a minute to register. The way you register is you go to login and you create an account right here on this form. After you've created the account, you will have access to the coursework that we will discuss. Okay, great, excellent. So I see that a lot of you already have this. And once you're logged in, you will be able to see, yes, you will be able to see some of the courses that are going to be included in this program. Okay, so is there anyone here that have not registered yet? Is there anyone here that has not registered yet? Okay, if you could take a moment and register, please. We'll give it just a couple of minutes so everyone is able to follow through some of the examples that we will be going through. Okay, so once you are logged in, you will be redirected to a page where you see a collection of courses that are linked to your profile. And what I will demonstrate is how we will leverage some of these resources to actually practice what we are going to do in this program. Okay, so I see that someone is raising their hands. Would you like to ask something? Okay. Okay, so if you're already inside on the course page, please search for data science, and you will see here a couple of courses: data science, introduction to data science in Python, and introduction to data science in R. Okay, so once you are in these courses, you will be able to see what types of methods we're going to be covering and even try some of them with a free registration. So if you register today or you already have been registered before, you can access all of the free courses. And then when you complete your registration, and there's a number of ways that you can register that I'll talk about, you will see courses with this star, and those would be premium courses that address some of the more advanced topics and require some more practice. So you'll be able to access those with a premium subscription. One of the things that is always difficult with large, complex data sets is to grasp the type of information or the type of relevant information that is in the data quickly, not spending a lot of time on individual feature like a gene, and more looking for general trends. So what I want to show you today briefly is an example from a course on dimensionality reduction, principal component analysis, and visualization. Now, principal component analysis is most useful for continuous data, which means that the data is in a numeric form, just like you see inside the cells over here. And it could be anywhere in a continuous scale from 0 to whatever the maximum is. And it doesn't even have to be 0. It could be also centered. So it could be -1 to 1, or it could be negative and positive numbers. But the key point here is that it is continuous data. And so the continuous data is represented here in this example as a gene expression profile for a number of different groups. As you can see right here, we have a row for groups, and we have sample IDs, which are essentially names of cell lines that have different properties. So they come from different breast cancer patients. And so using this example, what you'll see in this course is how you can apply a principal component analysis technique to quickly evaluate the separation of samples into different groups based on this gene expression profile that is found inside the data matrix, which is just the numeric portion of this table, of this data frame that we were looking at just a moment ago. So you have a brief video that you can go through to really understand the challenge of the process to getting to this outcome, but also you have a breakdown of the full code. Now, for many people, including myself, one of the main challenges in learning data science is that most of the courses out there start with very basic things like learning how to print out hello world. In our case, we're assuming that you are ready to jump beyond that and start with a data set that you understand. And that's why we've selected these data sets that come from a background relevant to life sciences. And so I think many of the people are more motivated to continue and learn about a method once it's talking about a data set that you understand. So in this case, this is gene expression for cell lines, and those cell lines could be categorized in groups based on their type or subtype of breast cancer. And so the first thing that you will see here are some important parts of the code that you have to master for any kind of a script. And in this case, we have different libraries that will be used. And these libraries include libraries which are packages that include many different functions that we can pull from to actually apply to the data set that we load. The second line here is going to be how to load this data. So this data exists here on the internet, but how do I put it into the script, right? And so that will be there. And then we start working with this data set to extract only the meaningful portion of the data for our analysis, and then launch the analysis. Now, once you learn these brief steps, it's very easy to understand how we can apply the same technique to many different types of data and how to start working with the tables. So here you see two different methods, the base method in R and the ggplot method in R for visualizing this data. And as you go through, some additional useful information is going to be added to the plot, like labeling the plot, or even projecting the feature significance or how individual genes contribute to this principal component analysis plot, a two-dimensional plot that we're looking at, so that we can evaluate which genes are actually responsible for the separation into different groups. So once you go through some of this basic explanation in learning the code, you will be able to try the code yourself. And trying the code starts from just simply pressing this button, run. Okay, so just by running this code right here in the browser, you already get some output. And you can play with this to change things so that you can see what is actually going on. For example, as you're going through this code, you can see that we can take there are actually two different methods to visualize the PCA output. So here you have the basic method with plot, and here you have a GG45, so a library to do everything in a different approach. And simply by deleting this, we can try to run it again and only get the basic plot, right? So now we can separate the plot into portions that we understand. So we understand what the first portion did, we understand what the second portion did. In order for me not to forget, I can just reload the full script and get all of the things that were in there before. So I can actually now remove this part of it right here, run it again, and only get the ggplot output, right? So now I'm working with two different libraries. One is the base library, including in R, and another one is a library called ggfortify that I can actually look up online and see what does it do, right? So I can start looking into all of the different options for ggfortify and see what does it do. And many of the experiences that you will have applying data science is going to be related to looking up different packages and seeing what kinds of additional methods are going to be used and how are these methods actually used by others to use, for example, for principal component analysis. So you can always find these packages on their different repositories online. Okay, a question from Abu Bakr. Can you run the code with your own data? Excellent question. So this is a code that is going to work anywhere, right? So simply taking this code right here and copying it to your clipboard, you can now install R and RStudio on your own computer and run this code on the data set that you would like to analyze. However, let's take a look at how this data was loaded. You can see that the input file is given via a URL. This is a URL to a GitHub repository where we store all of the data sets that we'll be going through. On your computer, that data set is going to be sitting somewhere else. So it's going to be in your folder, but in a very similar way, you're going to be using the path instead of a URL to a public domain. You will be using a path on your own computer. And you can do that by setting up a working directory on your computer and then linking to that directory, not through the full path, but just giving the name of the file. So that's something that you can try. And you can also try some of these downstream codes that are going to be much more detailed in what kinds of outputs they provide you with, ultimately leading to a interpretable result. Okay, so let's take a look here at the final result of this analysis. You can hover over the individual dots that represent samples, and you can see that they're colored in four different colors. There's the luminal, which is pink, basal, which is green, normal-like, which is blue, and cloud and low, which are dark blue. Now, what is this box that we can spin around? This is essentially a space defined by three principal components, the X, Y, and Z. Those are axes on in this space, or principal components selected by the top information that they carry based on how much percentage of variance they show for our data. And so just by learning about these types of outputs, we can start asking questions about similarity and differences between these groups of samples and how we can actually take a classification model to train an algorithm to identify the samples that we see here based on feature significance, based on specific genes that are in this data set. And so going step by step through examples that you can run right here on the screen to more and more and advanced methods, You can see here, going beyond the basics, we'll introduce some additional applications of statistics, for example, to understand confidence levels on this principal component, and eventually turning this into a data science problem. And the data science problem is going to basically tell us that I have a lot of data, I have mined this data, and I have identified some differences between groups that I can interpret clinically using phenotypic information. And now I want to address a problem of finding these types of subtypes in a different data set. And so now we can really translate this into an algorithm that will look for specific markers that will identify those types of patients or cell lines that match a certain clinical phenotype. So as a brief introduction to data science, I think this is a good example that shows us how we can apply this data-driven approach of data mining, classification, training a machine learning model, and asking the right kinds of questions to understand biology and to be able to leverage these signal points to really address a major problem, which is classification of different types of cancers, especially based on biomarkers. So genes that we can detect and we can then use on big data to mine those patterns. Okay, so if there are any other questions, you can go ahead and put those questions in the chat box. And in the meantime, I would like to return briefly to our presentation to talk about how the other components are actually going to be used. So as I mentioned, this is going to be a program that combines live sessions like we have today, where you can ask questions. We can go through code together and try to run it, maybe on our browser code playground, or maybe on your own computer. Compare the outputs and be able to ask specifically whether we understand them, with self-guided study materials. So materials that you can then practice online independently and be able to validate that you can actually go through these questions, go through the same types of challenges and examples, and then receive practical assignments. So in these sessions, we'll give you some assignments to complete. So you can try it, practice it, and then test whether you really understand them. Okay, so at this moment, I would like to take a pause and launch a poll so that we can get a better sense of who's in this session today. So, Sonanika, let me try launching the poll. Okay, so Sonanika, if you could please launch the poll. It seems like it's not possible from my computer.
Hi,
Sonanika,
yes, is the poll visible? It's launched from my side. Yes, I see people.
Okay, great.
Yes. So my first question is, what is your level of education, or in that matter, what is your designation? Are you a high school student, an undergrad, or a grad? Undergraduation, that means bachelor's, graduation, master's, post-graduation, or a PhD fellow or a postdoc fellow? And also, I would want to know about your research interests. So what are your research interests? Machine learning, data science, infectious diseases, or cheminformatics, genomics, transcriptomics, metagenomics, epigenetics, or the general bioinformatics? And also, we would want to know if you have signed up on the Omicslogic Learn portal. And if yes, it's great. If not, then I would be sharing the link with you, and you can go ahead with signing up and creating your profile. Thank you everyone for answering. I would keep the poll for more 30 seconds. Thank you.
Okay, thank you, Sonalika. And while you're completing that, there's just a couple more things that I wanted to share. Okay, so what I wanted to add on to this is that in the process of getting started with data science, but also getting to a level of understanding, as we already heard from Clinton, one of the highest levels of understanding is when you can meaningfully apply the methods and skills that you have acquired to a project where you can demonstrate not just that you've memorized some scripts, but that you actually understand how to extract meaningful signal, interpret it in biological terms, and be able to present it answering questions to experts and others, peers like you. So to do that, the Omicslogic portal incorporates several features that will help you do just that. The first one is that you will gain access to these technical skills that you can practice on given examples. But what if you want to try an example of a different kind? And so the first thing that I want to show you is that beyond just the technical coursework, you will also have access to project examples. And these include examples in cancer. They include different types of data from cell lines, from animal models, as well as from patients. That also includes some clinical information. You can also look for infectious diseases, where you will find information on COVID-19, Ebola virus, and others. You will be able to find many other types of examples that include agriculture, biotechnology, neuroscience, and many other examples. But beyond that, all of the methods and all of your activity on the site is geared towards allowing you to develop an independent project. And so to develop an independent project, I want to show you the profile that you will have, where all of the activity that you will be doing throughout the program is going to link up into an opportunity to track your own activity. So you can see where you are and how expert level you are reaching. You're also going to see your progress for the different courses that you might be interested in and taking. And then you will also be able to start an independent project. So the independent project will require you to complete your profile with a full picture and a brief bio about yourself. So profile picture and a bio. Then you will have to reach at least 10,000 points where you've demonstrated your skills and abilities by practicing some of the assignments and taking different types of examples from our repository of projects. And then finally, you will be redirected to a course that we've prepared on how to develop a bioinformatics research project. So here we will give you some practical guidelines on how to choose a topic for your project, how to find publicly available data. If you don't have your own data, you can take from the repositories that are available online, and then how to translate what you have identified as a topic and a data set into a real research question. And that research question will have a data component where you can apply your data science skills, but also will include a clinical, biotechnology, or pharmaceutical question, so a biomedical question that you can answer with this data. And so combining those two together, you will be able to design a project presentation and submit it for review by our team that is managing this platform. This process will culminate in a presentation that you will be able to share with others right under your profile. And to give you a sense of what these projects look like, you will see down here on the front page a few examples of participants from high schools, from undergraduate level of college and university education, as well as faculty that have completed projects in a very short period of time and were able to demonstrate how to apply the methods that they have learned to a data set and a topic of their interest, and how did they leverage it to publish, to present a poster session, to apply for jobs, or advance their research projects and careers. And so I think that is really the culmination of this program, is that we will be able to submit a project proposal, and if you choose to stay long enough, you will be able to complete this project and actually come out with a full project presentation at the end. So during this time, we realized that many of you are going to find it challenging processing big data on your laptop. And so what we are going to provide you with is a 5 terabyte storage space on our big data analysis platform, where you will be able to utilize big data analysis techniques to process genomic, transcriptomic, metagenomic, or epigenomic data, and try some of the tools in a user-friendly format. So even before you know any coding, you'll be able to understand how to process, visualize, analyze using statistical methods, and train machine learning models using this interface that you don't need any coding for. This platform combines many different types of data that you can try. It also provides access to tools using data mining. And importantly, this will help you practice and identify the area and the type of data that you would like to focus on. Now, it includes a simple to follow interface with explain methods, color-coded maps with different types of inputs and outputs that you can try, and provides you with a guided process that is both interpretable, reproducible, and following some standard applications and outputs that you can directly use in your final project. It covers things like sequence alignment, evolutionary studies using phylogeny, variant calling, RNA-seq, machine learning, and structure analysis. Beyond that, in the code playground, you will find tutorials on both Python and R, just like what we saw, and many of the tools that you've done on our visual pipeline builder would be downloadable as a code in R that you can right away open up and start working through on your computer. Now, some of the code is also going to be in Python, and so we will have a full session to introduce you to how to install and configure Python on your own computer, but also how to use some of the exercises to start building up that portfolio or library of useful scripts that you can utilize. So we already talked about PCA in R. You'll see the same is available in Python, and you will see that many of these methods are going to be applied to a variety of types of data, so that you can try genomic data analysis as well as transcriptomic, epigenomic, and metagenomic types of data. Now, we will talk about how do you learn the standard outputs, but also how do you then apply your data science thinking and solve problems, right? So it's not going to be only practicing some scripts, it's also going to be problem solving once you acquire some of those skills. And that will include finding the right piece of code from the code that you've already practiced, or even writing some of the code on your own so you can be more creative with what you're going to be doing. Okay, so sorry about the date here seems to be outdated, but we're going to cover this whole topic in 2 weeks. So these are essentially going to be 2 big parts of the program, and they will cover an introduction where we will learn about the methods and the way these methods are applied to specific examples, and then go through the practical coding skills that you need to continue working on these challenges. And as I mentioned, it will culminate in an independent project that will combine public domain data or data that you have on your own with methods that you have learned and that you have understood how they work to develop essentially a novel project proposal that will demonstrate your ability to work with different types of data and extract meaningful insights. And for that, we have a number of examples that we will introduce you to of how others have benefited from this journey, including some of the people that already work in research using some of these tools and using some of the skills that they have gained. So we have a number of different participants. They all have benefited from not only the research component of this program that allows you to really focus on research questions, but also from the technical component, allowing you to practice these skills and receive guidance from mentors to be able to solve challenging problems in biomedical research using data science. So for those of you that are interested to join this program, again, I recommend looking through some of the resources that we've already looked together at on this portal. You can always go back to the programs page right here and find the program is called LBRN Omicslogic Data Science. So this program is going to be starting on July 12th, so very soon. And if you have any kind of questions about this program or about how to register, we recommend joining our session like this. So we will have a number of webinars dedicated to this program, but also reach out directly to us. We have a marketing team at marketing@pine.bio. And if you have any questions about payment, registration, if there are any discounts available to different groups, depending on where you are located geographically, you can reach out to us with all of those questions and ask about certification, ask about the resources involved, or how to get in touch with mentors once you are registered for the program. Okay, so I think that should give a good overview of the program itself. If you do have any questions, we have a minute or so to answer some of the questions here. Or as I mentioned, you can easily reach out to us after the session as well, and we'll be happy to provide you with some of those answers. And Clinton, if you wanted, you can add anything else you would like to say add to this.
I don't have, you were very comprehensive. I don't think I have any other thoughts. I do think this is a good way to transition into a more analytical role. So that would be my only point.
All right, thank you, Clinton. And everybody will have a chance to get to know Clinton, who is a great guy and very knowledgeable in data science, in molecular biology, but also now in training. And so we're excited to have him on board and help out with this program. And we're excited for all of you to be able to join the sessions that we are planning to start in July. So if there are no other questions for now, we will finish the webinar, and we welcome you to explore the coursework, and do let us know if you're interested to join between now and July 12th, and have a great rest of your day. Thank you for joining, Clinton, Sonanika, Ojasvi, everybody else. Take care.
Thank you everyone for joining. Bye-bye. See you in the
Thank
session.
you.